we've been looking at the details of the chemicals that make up a double strand here of DNA. We're just going to look at a few different processes that DNA can be involved in now. So the first process that we're going to look at is replication, and that is when you copy DNA. So we have the parent strand up here, and then if I need to make a new cell, then I need to divide that or copy it into two daughters. So I end up with 2 copies instead of 1 copy, and now I can divide my cell. So we're not going to go into great detail on this. We're not even going to talk about the enzymes that help with this. Although looking at the enzymes, DNA polymerase, you can tell kind of what it's going to be used for. But just a couple things I want to show you here, okay? Remember what held my base pairs together? It was just those intermolecular forces. And so that allows me to unzip my structure pretty well in replication by just breaking those intermolecular forces, which an enzyme helps to do. So I break those intermolecular forces, and then I start to form new ones as I build my new daughter strands here. So here I break them, and then here I reform those intermolecular forces in the daughter strands. So that is one little thing to look at. Again, you can see that we would have the same pattern where here are my base pairs that are getting split up. So I've got lots of base pairs still here, and then my backbone would have sugars all along it on both sides, and I'd have those phosphodiester bonds holding the sugars together. So it's the same exact patterns that we saw all the way back here at the beginning, right? Phosphoester, sugar, base, phosphate, sugar, base. We can see that here. It's just kind of unraveling it a bit. So with replication, we are still going to always end up pairing our bases up. We have to have opposite directions for our DNA strands. So when I make complementary strands of our DNA, I'm going to have the opposite direction. So it's going to go from 3' to 5'. And I just want to match up those letters like we saw on the very first slide today. So C, cytosine, always pairs with G, guanine. G pairs with C, cytosine. T pairs with A, so that's thymine with adenine. And then A pairs with T, G, C, T, G, A. So this would be, as I build my 2 strands here, if I have here a C, GT, then I'm going to have a GCA come in to complement it on the other side. So replication is kind of interesting also for not just what goes right, but also what goes wrong, and there's so much more depth than what we're going to go into here. But high energy radiation, such as ultraviolet or X-rays, can cause mutations as this whole process is happening. And maybe you've heard about those mutations even with the coronavirus that's happening right now. They can predict like roughly how often mutations happen, and mutations can sometimes change the virus to make it more active or the same activity, but they keep an eye on these mutations. So one of the places those can come from is just random mistakes, but you can have extra mistakes if you expose it to high energy, which is not unlike life, right? If you have too much energy, you do things too fast, you're more likely to make a mistake. So that's kind of what's happening here. You can take advantage of that if we want to kill DNA or destroy it. So radiation therapy used for cancer is such a high dose of high energy radiation that the DNA damage causes cells to die. So if you are looking to kill your cancer cells, then you target them with a lot of radiation, and that causes so many mistakes here that you can't go on and divide your cell. So radiation is one way to tackle and kill cancer cells. Another way is chemotherapy, and for most cancers now you want a combination of both of these. Chemotherapy, I'm reading a book right now called The Emperor of All Maladies. It's the history of cancer. Super nerdy, but super interesting. But it talks a lot about the history of chemotherapy. It's really been just in the last really 20 years they've gotten any better at it, and they just threw chemicals at people for decades now to see if any of them would help basically stop this replication from happening. So both of these are going to destroy the ability of a cancer cell to replicate. So every time we're dividing, cancer cell needs to keep dividing, right? But a chemotherapy drug can interfere with replication. So again, it's not a bio class, so we're not going to go into this in great detail, but we can interrupt some of the base pairs here. We can close up the base pairs. We can prevent our DNA from unwinding. We can also interrupt this separation where you move the DNA here. So there's a few different approaches that we can take to interfering with DNA replication. And so what chemists would do is work on chemicals that maybe match up perfectly with or very similar to the chemicals involved here, and then they can get in there and interrupt that replication process. So just kind of an interesting angle to take on our DNA. So replication is going to be this copying process. You need to be able to write that complementary strand and understand just a tiny bit about the two different ways that we can mess up this replication.